Hello students. Today this one is very important chapter, right? Molecular basis of inheritance. Molecular basis of inheritance, right? In this chapter we have to read the structure of DNA because it is a genetic material, search for genetic material, RNA one, replication, transcription, genetic codes, then after translation, regulation of gene expression. Here lack of purine we have to read, right? You understand here DNA, structure of DNA because it is genetic material. Search for genetic materials means here experiments which are done by different scientists, right? RNA one, then replication is DNA forms its copy. Transcription is formation of messenger RNA. Genetic codes, how many genetic codes? 64 genetic codes. Translation means protein formation. Regulation of gene expression, here only we have to read this lack of purine model. Human genome project, only one genome project this time, it is only human genome project is there, and DNA fingerprinting, right? Today we are going to this DNA structure. In this chapter what we read about the structure of DNA. In the previous chapter means it is our inheritance, right? You have learned the inheritance pattern, how genetic information comes into the next generation, means in offspring, and the genetic basis of such pattern, right? At the time of Mendel, because Mendel time, what already we know, at the time of Mendel no genes, genes term is not used at the time, right? So he used this factor. The nature of those factors, today we set them genes, regulating the pattern of inheritance was not clear. At the time, this one is not clear. Chromosome is also not known by us, right? Over the next 100 years, the nature of putative genetic material was investigated, culminating in the realization that the DNA, that is deoxyribonucleic acid, is the genetic material, right? At least for the majority of organisms. It is given at least for the majority of organisms. That means what? There are some organisms in which RNA is also genetic material. As for example, RNA viruses, right? They have the genetic material of it is RNA, not DNA. But when we consider from that is bacteria, Monera to Animalia kingdom, their genetic material is only DNA. In class 11th, you have learned the nucleic acid as polymer of nucleotides. What is nucleic acid? So here it is given the definition of nucleic acid. Nucleic acids are the polymer of nucleotides, right? And nucleotide means what? It is the combination of that is our nucleoside plus phosphoric acid, right? When phosphoric acid attach with nucleoside, then it forms a nucleic acid, right? Then we use word acid, okay, due to the presence of phosphoric acid, H3PO4. Deoxyribonucleic acid, DNA, and ribonucleic acid, RNA, are the two types of nucleic acid. We know only two types of nucleic acid is there, RNA and DNA, right? Found in the living system. DNA acts as genetic material in most of the organism. Genetic material is DNA because it's stable structure, right? RNA, though it also acts as genetic material in some viruses. Which viruses? Plant viruses. We can say plant viruses, right? Plant virus and viroids also. There is genetic material is RNA. As for example, tobacco mosaic, TMV, tobacco mosaic virus. Not only plant virus, some animal virus like AIDS is also RNA virus. This time a tragedy came, corona. So coronavirus is also a RNA viruses, right? Mostly function as a messenger RNA. Means RNA, it acts both messenger RNA or it acts as genetic material also. RNA has additional role as well, right? It function as adapter, as for example transfer RNA, structural, as for example ribosomal RNA, and in some cases as a catalytic molecule. Catalytic molecule means in ribozyme, right? Which RNA act as that is catalyst? it is, we can say it is 23 sRNA, right? In prokaryotic it is 23 sRNA and eukaryotic it is 28 sRNA. Okay, catalytic molecule ribozyme. In class 11, we have already learned the structure of nucleotides. Already we read about the, but not detail. Okay, I know in class 11th, nucleotide structure is given, but not in detail. Today I am going to tell you about these structures, right? Nucleotides and the way these monomer units are linked to form nucleic acid polymers, right? Monomer units means nucleotides. How many nucleotides are there? We know we have how many nucleotides are there. In case of DNA, 4 nucleotides. In case of RNA, also 4 nucleotides. But when we consider only nucleic acid means how many nucleotides are there? There are 5 types of nucleotides. Which one is adenylic acid, guanylyl acid? These are purines. Then pyrimidine, cytidyllic acid, thymidyllic acid, or uridylic acid. These 5 types of nucleic that is we can say nucleotides are there. These are nucleotides. Okay. In this chapter, we are going to discuss the structure of DNA, how a DNA is formed, its replication, how it replicates. We know replication occurs in which phase of the life, in which phase of the cell cycle. You can say, I know, you know, it is the S phase, right? In synthesis phase, S phase, DNA replicates. So, how it replicates, we read in this chapter. The process of making RNA, means first transcription, how it transcribes the messenger RNA, means how it gives code in messenger RNA from DNA, right? That is called transcription. And the genetic code that determines the sequence of amino acid in proteins. Then this genetic code, right, it is responsible for the formation of protein. Protein means what? A specific sequence of amino acid is prescribed by the DNA in RNA, right? That is what determines the sequence of amino acid in proteins. Who determines this sequence of amino acids? It is the genetic code, which is provided by DNA in messenger RNA, right? That process is called protein synthesis, and this protein synthesis process is called translation, right? Translation and elementary basis of their regulation. Also, gene expression means regulation. How they regulate? Means 4 levels are there, or gene expression regulation. We have 4 levels. We will read this one, right? 4 levels means what? Okay, I tell you here. Otherwise, you get confused. 4 levels means first level is transcription level. Second means what? It is the transcription. Then after what happens? This after transcription, messenger RNA is formed at splicing level. Transcription level, then splicing level. At third one is where it is moves from nucleoplasm to cytoplasm, because this messenger RNA, it has to move. Either messenger, transfer, or ribosomal RNA, they come outside the nucleus and they come into the cytoplasm, right? So when they move from nucleoplasm to cytoplasm, that is also a stage where this expression is regulated. And last, at the translational level, when this messenger RNA forms a protein. These are the 4 levels where these which one? Expression of gene or gene expression is regulated, right? And the determination of complete nucleotide sequence of human genome during the last decade. Now here in the last, what we read here, the complete nucleotide sequence of human genome, nitrogen base sequence, base pair sequence of a human genome during the last decade. Last decade means it is because 1990 to 2003, 13-year mega project is there, which we read in this chapter, right? Mega project of that is human genome, right? Project. Also we read asset in the new era of genomics. And okay, the time it is 30 years. Mega project is there, but it is it finished in May 2006, right? Is 3 years late. In the last section, the essential of human genome sequencing and its consequence will also be discussed. Let us begin our discussion by first understanding the structure of the most interesting molecule. most interesting, we can say most wonderful, or we can say it is a responsible molecule for living beings, right? In the living system, and that is the DNA, deoxyribonucleic acid. In subsequent section, we will understand that why it is the most evident genetic material. Okay, we know because it is a stable structure, but we will read different properties for genetic materials and why DNA is genetic material, not RNA. Mostly DNA is there. Okay, RNA is also there. Some exceptions are there because RNA in some viruses that we can say retroviruses or RNA viruses, they have genetic materials. RNA is there, but only in viruses why DNA is formed due to the stability. Okay, and what is the relationship with its relationship is with RNA and okay, DNA. What is the relationship? Because we know here DNA for expression of gene, DNA is totally dependent upon RNA because RNA is capable for the formation of protein. DNA cannot form the proteins. DNA cannot express itself. For expression, DNA is totally dependent upon RNA. Okay. Okay. Now the first point is here the DNA. Right. What is the structure of DNA? First here, DNA is a long polymer of deoxyribonucleotide. Long means very long polymer, right? It is a macromolecule. The length of DNA is usually defined as the number of nucleotides. Number of nucleotide, that is, or a pair of nucleotide referred to as base pair. Base means what? Nitrogen base pair. Why it is defined as a number of nucleotides? Because we know the distance between two nucleotides is 3.4 angstrom, or we can say 0.34 nanometer. Right? Unit, if we use this unit nanometer, 0.34, because nanometer means what? 10 power 9 meter, and angstrom is 10 power 10 meter. So the distance between two nucleotides is 3.4 angstrom or 0.34 nanometer. If one nitrogen base pair or one base pair, or we can say one nucleotide is attached, means 3.4 angstrom length is added. One is removed, that means 3.4 angstrom length is removed. The number of base pair, they determine the length of DNA. We should, how we calculate the length? We have to multiply number of base pair multiply by 3.4 angstrom or 0.34 nanometer, then we get the exact length of genetic material. Here it is, simply usually defined as the number of nucleotide or a pair of nucleotide referred to as the base pair present in it. Correct. This also is the characteristics of an organism, because every organism have different type of not different types. DNA is same but different length, right? Means nitrogen base pair, their number is also different. So it is the characteristics of every living beings. For example, a bacteriophage phase known as 5×174 has 5386 nucleotide. What is the length of this one? Multiply by 3.4 angstrom, then we get the length. You have to read this one. How many nucleotides are there? 5386 base pairs are there, and when we multiply this one with 3.4 angstrom, we get the exact length. Another example is bacteriophage lambda. Bacteriophage lambda, it has 48502, we can say 48,502 base pairs are there. When we multiply this 48502 with 3.4 angstrom or 0.34 nanometer, we get the exact length. Base pairs are there. E. coli, Escherichia coli is a very important bacteria, right? E. coli, it has how many base pair? Here it is given 4.6 into 10 power 6 base pair, right? Simply we can get the length multiplying by 3.4 angstrom, right? And haploid content of human, here it is given haploid content, not diploid, right? Haploid content of human DNA is 3.3 into 10 power 9 meter, 10 power 9 base pair, right? It is haploid. Diploid means what? Diploid means 2n. 2n means what? Multiply by 2. So in diploid we can say here 6.6 into 10 power 9 base pair. Haploid 3 into 3 into 10 power 9 base pair. Right? So this is the human DNA structure. Let us, when we multiply this 3.3 into 10 power 9 base pair multiply by 3.4 angstrom, then we get near about 2.2 meter long polymer of DNA in human. Right? So this is not 3.3, we can say here 6.6 because here the diploid content of human, each cell, we are diploid animals, right? So means it is haploid, it is for haploid, only for gametes, right? In our cell it is 6.6 into 10 power 9 base pair. When we multiply this one with 3.4 angstrom, we get the near about 2.2 meter long DNA molecule. That is genome, total genome, not one chromosome. It is a total genome, right? Let us discuss the structure of such a long polymer. Now we are going to discuss the structure of these long polymers, right? The structure of polynucleotide chain. Before going to there, I want to tell you about the, we can recall the structure of purines and pyrimidines. So see first here. Now first question, when we go to the structure, first what are the component of DNA? First here, component of nucleic acid, simply I write here, nucleic acid. So when we see the component of nucleic acid, we have three components. Three components means, how many components are there? Three components. Which one? First is our nitrogen base, right? Second is our sugar, and third is our phosphoric acid. Okay, three components are there. This nitrogen base, it is further divided into two parts. We can say purine and pyrimidine, right? This sugar, which type of sugar is there? It is pentose sugar. Pentose sugar, and this pentose sugar, it is also two types. One is ribose and another one is deoxyribose. And this phosphoric acid, it is only H3PO4. Okay, these are the component of nucleic acid. So first you can see here phosphoric acid. So phosphoric acid, structure of phosphoric acid is this is the central molecule, right? One side double bond O, here it is OH, OH and OH. This is the structure of phosphoric acid, H3PO4, right? Now second is our sugar molecule. Which type of sugar is there? It is pentose sugar. Pentose sugar means what? Number of carbon is, this number of carbon is C5. That's why it is called pentose sugar. So C5, H means what? 10, because it is a monosaccharide. C5, H10O5. That is ribose sugar. But when we consider the deoxyribose, it is C5, H10O4. One molecule is less in this deoxyribose, means second carbon oxygen left, right? Oxygen of second carbon is absent. Okay, purine and pyrimidine, this purine is double cyclic and pyrimidine means what? Single cyclic. Okay, now second it is sugar. Now I am going to tell you about the structure of sugar. You can see here. Sugar. Sugar means which sugar? It is a pentose sugar. Pentose sugar. Pentose means 5 carbon. 5 means we know here CnH2nOn. We use this formula because the monosaccharide, right? Or we can say here, I already tell you, CnH2n H2ON. That is the formula of monosaccharides. Right? Okay. Now because it is a pentose sugar, means number of carbon is 5. So number of carbon is 5. Here it is ribose sugar. Example is ribose. Right? Its structure is C5. Then H means what? It is 10. O means what? N means what? 5. This one is ribose sugar. What is the structure of this ribose sugar? Here we can say oxygen, first carbon, second carbon, third carbon, fourth carbon, and we know fifth carbon is always outside the cell. Here it is CH2OH. This one is CH2OH. Right? This side it also OH, H. This one is H, OH. Also here OH, H. Right? So this one is the pentose sugar. This one is ribose sugar. First carbon, second, third, fourth, and fifth carbon. Fifth carbon is outside the cycle. We know very well in monosaccharides this time there is pentose sugar, fifth carbon is outside the cycle. If it is a we can say it is a glucose molecule, what happens? Hexose sugar, last carbon is always outside the cycle. This is ribose, and when we consider the deoxyribose, here the molecular structure means we can say the formula of deoxyribose is C. It is also a pentose sugar, but H10OH4. OH4 means one oxygen is less than ribose sugar. So which carbon lack the oxygen? This second carbon lack the oxygen. Now we can say here if we draw a structure like this one, oxygen, first carbon, second carbon, third, fourth, and this one is CH2OH. Right? CH2OH, H. That side also HOH. This one is H and H. No OH is there. Right? This side it is OH and H. Right? So here what happens? What we see here? Here we can see here. Right? Look, this one is right. Now this second carbon, look here, it lack oxygen. Because it is the first, second, third, fourth, fifth carbon. Because second carbon lacks oxygen, that's why it is called deoxyribose sugar. Right? Second carbon, and this is the responsible for most stable structure. Here OH is there, means it becomes, means this ribose sugar become active. Why it is more active? Due to the presence of this hydroxide, means OH oxygen at means OH group at second carbon. Due to the presence of OH group at second carbon, it is more active. This ribose sugar is more active than deoxyribose sugar, and it is responsible for this less lack of oxygen on second carbon is responsible for the stability of DNA. We will read this one after that. Right? Okay. Third, these are the sugar. Now third we have nitrogen base. Nitrogen base, which is present in our nucleic acid. We know nitrogen base is two types. First one is purine. Right? So purine, we have two purines. Which purine? First one is our adenine, and second is our guanine. Adenine and guanine. Right? Adenine and guanines. They are double cyclic structure. Both are double cyclic structure. I already tell you, right? Now it is represented by capital A, and this one is represented by G. Second is our pyrimidine, pyrimidine means single cyclic structure, single hexagonal cycle is there. This one is one double cyclic, means one is hexagonal, another one is pentagonal, right? Now pyrimidine, three types of pyrimidines are there: cytosine, second one is our uracil, and thymine. 3 C, represented by C, U and T. Okay. Base structure of purine is hexagonal and pentagonal structure, and base structure of pyrimidine is only single cyclic, that is hexagonal. This is the base structure, right? Okay, now we are going to see the different structure of adenine, guanine, cytosine and uracil, right? First is our purines. Here it is adenine. Adenine means amino group, right? And guanosine, when ketone produces, ketone means what? Double C, double bond O is present, right? You can see here. First I draw here adenine, this side I write guanine. Base structure same for both. This one also base structure is same. Look, both base structure is same. What is the difference? Adenine, it contains NH2, but here guanine, it contains double bond O. Right? What is the location of nitrogen? We can say here this nitrogen and nitrogen. Same place, nitrogen and nitrogen. Right? Numbering means nitrogen is present in 1, 3, 7, 9 at this place. So 1, 3 means what? This is the first. 1, 2, 3, 4, 5, 6, 7, 8 and 9. Same 1, 2, 3, 4, 5, 6, 7, 8 and 9. You can see here what is the location of 9 or location of nitrogen? It is 1, 3, 7, 9. Remaining part means carbon is there. Here also carbon. This side carbon and carbon. Same carbon and carbon. Now when you see the valence, then you understand what the structure. Here we know very well. Hydrogen means 1 valence, oxygen means 2, then after nitrogen 3, carbon 4 and phosphorus 5. So remember this one. This is the atom and this one is the valence. 1, 2, 3, 4, 5. Hydrogen 1. Now you compare this valence and you get the structure, specific structure of these molecules, right? Now can you tell me the structure here? Let us consider here. Carbon. Nitrogen means 1, 2, 3 is completed. Carbon is 4. We need 4. 1, 2, 3. We need one more. 1 draw. Now 1, 2, 3, 4 valences of carbon is completed. Nitrogen also 1, 2, 3 is completed. Right? Now this is the carbon. Here, what is here? Here we look here. 1, it needs 4 valences. 1, 2, 1 add 3 and one more we need. What happens? I draw here H. Completed 1, 2, 3, 4. This side 1, 2, 3, 4 carbon is completed. Nitrogen 1, 2, 3 is completed. 1, 2, 3, 4 completed carbon. 1, 2, 3 completed nitrogen. This carbon 1, 2, 3, 4 completed. This carbon 1, 2, 3, 4 is completed. That side nitrogen 1, 2, 1 more, 3 is completed. 3 valency of nitrogen is completed. Now this is the C. C I write here 1. Because it also need 1, 2, 3, 4. 1, 2, 3 and 4 is completed. Nitrogen 1, 2. We need one more. That is H. Nitrogen completed. Right. Do you understand here how this structure is formed? So this is the structure of adenine. This is the structure of adenine. You understand here? Now in guanine, how many nitrogens are present in adenine? We can say 1, 2, 3, 4, 5. Means we can say 5 nitrogens are present in adenine. And in guanine also 5 nitrogen is present. Here also 5 nitrogen is present. Same. What is the location of nitrogen? It is also 1, 3, 7, 9. Location of nitrogen. First, third, 7th and 9th. Now structure. Look, this one is carbon. 1, 2, 3, 4. Completed. 1, 2. We need one more. Add 1 H. Now it is completed. 1, 2, 3. Right? Now carbon. We need 2 more. 1 is here. Now 1, 2, 3. We need one more. Here NH2. Now you can see nitrogen is also completed here. NH2. CNH2 here. Right? Now nitrogen 1, 2, 3 is completed. Carbon 1, 2, 3, 4 completed. Carbon 1, 2, 3, 4 is completed. We need not any valency. Here nitrogen is there. Same process here. 1, 2. We need one more. Now it is also become 3. 1, 2, 3 is completed. Carbon 1, 2, 3. We need one. We add H. It is completed. 1, 2, 3, 4. Nitrogen. Now we need H. Here 1, 2, 3 is completed. So this is the structure of adenine and guanine, that is purines, which are heterocyclic structure. Guanine means number of nitrogen is 5. Adenine also number of nitrogen is 5, but location of nitrogen is different. Here this sixth carbon, here it has oxygen, but in sixth carbon it has amino group. Sometimes you forget. A trick is there. You cannot forget why. What is the means? Amino means amino group. Adenine, A for adenine, A for amino group. Right? And here ketone group is there. Right? First thing, A come first, N come first. Look here. A come before the G. Here N come before the O. Do you understand what I want to tell you? Means always remember, adenine means amino group and guanine means their ketone group or oxygen is there. So you understand this structure. Look, another. This ninth nitrogen always binds with first carbon of pentose sugar. Ninth nitrogen of both cases, adenine and guanine, this ninth nitrogen, it binds with first carbon of pentose sugar. Last nitrogen, first carbon. Last nitrogen, first carbon. Okay. Now second, purine. After purine, we have pyrimidine. So see the structure of pyrimidines. Single cyclic structure, we have 3 pyrimidines. First one is our cytosine. Second, uracil. And third one is our thymine. Thiamine or thymine we can write, right? Thymine. 3 pyrimidines are there. What is, because they are, what is the base structure? And single hexagonal structure. 1, 2, same structure, right? And 3. Base structure is same. Base structure is same. One thing, always we know adenine it binds with thymine, and cytosine, guanine, it bind with cytosine. That means both these are complementary, right? So here, because this is cytosine, and we know cytosine binds with guanine, Guanine, oxygen is there. That's why here it is amino group. Right? To understand this side, because thymine binds with adenine, so just complementary means it has oxygen group. Just opposite, just complementary group is present here. What is the location of nitrogen? That is the location of nitrogen. 1 and 3. Same in all these three. Understand? Any doubt is there? No doubt. Okay. Now another similarity in all these three. This is the double bond O. Double bond O. Same double bond O. It is present in all these three cases. It is same. Okay. Now see the balance. Because this one is carbon, 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 and carbon. Here also carbon, 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 and carbon. This side also carbon, 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 and carbon. Right? So see this one. Carbon is 4. 1, 2, 3. We need one more. Here 4 is completed. Right? Nitrogen 1, 2, 3 is completed. Carbon 1, 2, 3, 4 is completed. Nitrogen 1, 2. We need one. That is H. The active one. C means 1, 2, 3. 1, 2, 3. And one more 4. Here also it is H. Now 1, 2, 3, 4 is completed. So this structure is cytosine. Completed. Completed. Any doubt? No doubt. Completed. Okay. Now uracil. Because here 1, 2, 3, 4 completed. Then nitrogen 1, 2. We need one. That is H. Right? 1, 2, 3, 4 completed. Nitrogen 1, 2, 3. We need one. H. Completed. 1, 2, 3. H. 1, 2, 3. 1. H. Completed. That is uracil. This is the structure of uracil. This thymine. Thymine is also called as 5-methyl uracil. Right? Methyl, because methyl group is present here. In thymine, methyl group is present. Right? Okay, you can see here how methyl group is present here. Carbon 1, 2, 3, 4, nitrogen 1, 2, we need one. Same structure. All the structure is same structure in uracil and thymine, but one is difference here. Look here, nitrogen 1, 2, 3, carbon 1, 2, 3, 4, nitrogen 1, 2, we need one H. Now it is 3, 1, 2, 3, nitrogen valency completed. 1, 2, 3, carbon valency, we need one H. Completed. 1, 2, 3, but here it is not H, but it is CH3. You understand? Now look, 1, 2, 3, 4, 1, 2, 3, 4 completed. This one is thymine. That is the structure of thymine. Right? You understand the structure of this nitrogen base. If you have any doubt, you can ask. I think you understand. Okay. Now we come. As I already tell you, what is the location of nitrogen in case of pyrimidine? Location of nitrogen is, look here, when we this one 1, 2, 3, 4, 5, 6. Same is there. 1. Like this one is 1. 1. This one is 1. 1, 2, 3, 4, 5, 6. Right? That side also 1, 2, 3, 4, 5, 6. What is the means here location? It is 1, 3. Right? Another thing. Which nitrogen binds with which nitrogen binds with pentose sugar? It is the first carbon. First nitrogen bind with first carbon of pentose sugar. Here first nitrogen, this is the first nitrogen, it binds with pentose sugar. And in pyrimidine, in purine, whatever last nitrogen, that is 9th nitrogen, it binds with first carbon of pentose sugar. Okay, so remember this one. This is the structure of, these are the structure of nitrogen base. Now here we read about the structure of nitrogen base, pentose sugar and phosphoric acid. Once you can see here, these are the pyrimidine. This one is our purine structure. Here, this is the base structure of purine and pyrimidine, and here this is the structure of sugars, both sugar, ribose and deoxyribose. Here you understand deoxyribose means second carbon, oxygen is absent, and in this page you can understand the structure of phosphoric acid. Okay. Now, what is the structure of nucleoside? First here, what do you mean by nucleoside? Nucleoside means what? That is our nitrogen base plus sugar. Sugar means what? Pentose sugar. That is called nucleoside. How many nucleosides are there? Because we have 5 types of nitrogen base, so we have also 5 types of nucleosides, we can say. Yes. Okay, when we consider sugar, we have 2 types of sugar are there, deoxyribose or ribose sugar. We have also, means when we consider sugar, we have 8 types of nucleosides. As for example, adenine plus deoxyribose, it is called as adenosine, deoxyadenosine or adenosine, right? Deoxyadenosine, DNA adenosine, and RNA oxygen means it is adenosine only. So here, when we consider that is nitrogen base plus pentose sugar, suppose that here adenine is there, adenine plus ribose sugar, then what is the nucleoside's name is adenosine. Second, if guanine plus ribose, it is called as guanosine. Simply, so sine, you can understand here these are purines, and for purine what we use? Sine. Suffix is sine. It is the suffix. We use suffix sine for purine, right? Second, we have cytosine plus ribose sugar. Now it is called as cytidine. Cytidine. Dine come from pyrimidine, right? Third one is our, suppose uracil plus ribose, it forms uridine. Dine come from pyrimidine, right? That here suffix dine is used. It is thymine plus ribose, it is called as thymidine. And these are all, they are pyrimidines, right? Here which suffix is used? Dine. Dine suffix is used, that is suffix dine for pyrimidine, right? So these are nucleosides. These nucleosides are RNA nucleosides, because here what we use, sugar is ribose sugar, right? Same thing is there. If we use, look here, this is thymine. Thymine is present only in DNA. This one is present only in DNA. So ribose sugar is not attached with thymine, but here which sugar is attached? It is deoxyribose. So it is better that we use here thymine plus deoxyribose. If deoxyribose I write here, that which nucleoside is formed here? Deoxythymidine is formed. So we should write here deoxythymidine is formed, right? If uracil only ribose is there, no deoxyribose binds with this one, right? Cytosine, if cytosine plus deoxyribose, it is called as deoxycytidine. Guanine deoxyribose, deoxyguanosine. Adenine deoxyribose, deoxyadenosine. Do you understand, right? So we can say here these 4 They are these four. They are the RNA nucleosides because they are binds with ribosome. If they are binds with deoxyribose, so they forms deoxy. That means we can that are DNA nucleoside. What is the name of the that is DNA nucleoside? So simply we add deox, right? Deoxyadenosine, deoxyguanosine, deoxycytidine, deoxythymidine. Because uracil is present. This uracil, it is present only and only in RNA. Only RNA, right? And this thymidine, it is present only in DNA, right? Now, can you tell me the number of deoxy or that is RNA nucleoside and DNA nucleoside? We can see here 1, 2, 3, 3 2 6, 7, 8. 8 type of nucleosides are there. Adenosine, deoxyadenosine, guanosine, deoxyguanosine, cytidine, deoxycytidine, uridine and deoxythymidine. Do you understand? Right? Okay, now this is the nucleosides. Now nucleotides. After this discussion, I will tell you about the structure of nucleotide and nucleosides, right? The nucleotide, same as to nucleoside. How many nucleosides are there? That is the number of nucleotide also, right? Nucleotide means what? We can write here nucleoside plus phosphoric acid and they forms nucleotide. Okay. Nucleoside means what? If we this one, we have nitrogen base plus sugar, pentose sugar plus H3PO4 and it forms nucleotide. Okay. Now, here I use this nucleoside. Already we know the structure of this combination of nucleoside, right? So, which nucleosides are there? We have adenosine, first adenosine, second guanosine, third we have cytidine, fourth we have uridine and fifth we have deoxythymidine, right? It may be deoxyadenosine, deoxyguanosine, deoxycytidine. If we consider the DNA, means that is our deoxyribose, so it becomes deoxyadenosine, deoxyguanosine, deoxycytidine. Uridine means only uridine is there because it is present only in RNA. This is present only in DNA. This is only in RNA. It is only in DNA. That is only one form is there, deoxythymidine, and uridine means only one form is there, uridine. And these three, they are both, you can say, they are both means what? In RNA and DNA. They are present in both. They are present in both. Okay. Now, adenosine plus H3PO4, it forms adenylate acid. Guanosine plus H3PO4, it forms guanylic acid. Cytidine plus H3PO4, they forms cytidyllic acid. We use dilylic here, suffix. Here nylic, that's a dilylic acid, right? Uridine plus H3PO4, that is phosphoric acid, it forms uridyllic acid, right? And deoxythymidine plus phosphoric acid, it forms deoxythymidinic acid. You understand here which suffix is used here? Here we use the suffix -ilic suffix for purines, because these two are purines, and for pyrimidines we use dialic acid. Dialic suffix, nilic suffix and dialic suffix. For pyrimidine, dialic. For purines, it is nilic. Do you understand? Another name is also there. This one is also called as AMP. Shortcut is GMP. This one is CMP, UMP, and TMP. Shortcut, that is adenosine monophosphate. Only one phosphoric acid is there. One means mono. Adenosine monophosphate. This one is guanosine monophosphate. That is cytidine monophosphate, uridine monophosphate, deoxythymidine monophosphate. Okay, when adenosine means plus H3PO4, it is called adenosine monophosphate. But when it is a DNA adenosine, means we can say deoxyadenosine. So it becomes deoxyadenosine monophosphate. This phosphoric acid, it is attached with the fifth carbon of pentose sugar, or we can say last carbon of pentose sugar. Last carbon means fifth carbon. So because it is attached with fifth carbon, so here what is the full form of this AMP? It becomes adenosine 5-monophosphate. Adenosine 5-monophosphate. Same, guanosine 5-monophosphate. That is cytidine 5-monophosphate. Uridine 5-monophosphate. Thymidine 5-monophosphate. Deoxythymidine 5-monophosphate we use. Why 5-? 5- indicate that this phosphoric acid is attached with the fifth carbon of pentose sugar. Do you understand? Right? Adenilic acid, we can also say this one AMP, adenosine monophosphate. Same, you know, this AMP, when it also become ADP, AMP, ADP, AMP for adenosine monophosphate. When it convert into energy coins, that is ADP. It is called as adenosine diphosphate, adenosine 5-diphosphate. When it forms energy currency, that is ATP, it is called as adenosine 5-triphosphate. 5- means phosphoric acid is attached with the fifth carbon of pentose sugar, right? First carbon of pentose sugar, it is attached with the nitrogen, ninth purine. In case of purine, ninth nitrogen, and in case of pyrimidine, first nitrogen of nitrogen base. First carbon, last carbon. First carbon, fifth carbon. Fifth carbon, phosphoric acid. First carbon, nitrogen base. Okay. Now we are going to see the structure of nucleoside and nucleotides. Of nucleoside. First, the structure of nucleoside. Here I tell you one structure of purine and one structure of pyrimidine, and at home you have to draw the structure of other, means purine or pyrimidine. Okay. That is, suppose that I am going to draw a structure of adenosine. Okay, you can decide which structure I made here. Which structure? Okay, adenine, adenosine. Okay, adenosine. Here we are going to draw a adenosine, one nucleoside, that is adenosine. Adenosine, because it is a purine, adenosine. Adenosine, because adenine is a purine, it is double cyclic structure. So we draw here base structure of adenine, double cyclic structure, right? And we know also here it is the position of nitrogen. Okay, because it is a adenine, so we know here it is NH2 amino group is there. That side 1, 2, 3, 4 carbon. This is the carbon, right? This is the carbon. Here it is, what is there? It is H 1, 2, 3 N, right? This carbon, carbon, nitrogen, carbon, H, and this is the H. So this one is the structure of, this is the structure of adenine, nitrogen base. Now, when we draw the nucleoside, so here pentose sugar, oxygen, first carbon, second carbon, third carbon, fourth carbon, and last carbon is outside the cycle, right? First carbon, it has OH and H, right? H, this one is H and OH, sorry, OH and H, right? You can see here, this OH and H, they bind with each other and one water molecule is released. Now, a bond is formed between C and N. One bond is formed between C and N, and this bond is called glycosidic bond. Because carbon binds with nitrogen, so we should write here N-glycosidic bond. It is called N-glycosidic bond, right? For the formation of one nucleoside, one water molecule is released and one N-glycosidic bond is formed, right? Because N last nitrogen binds with first carbon. Look here, this is the first carbon, first, second, third, fourth, and this is the fifth carbon. And here it is the ninth nitrogen, right? We can write here 1, 2, 3, 4, 5, 6, 7, 8, 9. Do you understand? This is the structure of adenosine. Okay, one thing, if we draw the structure of guanosine, so simple, all the structure are simple, right? You have to change this NH2 in double bond O, and this side, this second carbon, it is attached with NH2 amino group. That is the difference. All these things are same. Same N-glycosidic bond is formed, one water molecule is released, right? Second structure, it is the purines. We can say adenosine means purine. And when we consider the pyrimidine, suppose that one structure, this is the adenine. Suppose that I am going to draw a adenine, just opposite, just complement of adenine is, we know, just complement of adenine is thymine, right? If I draw a thymine means thymidine structure, because we are going to draw the structure of nucleoside. Nucleoside is thymidine, right? Thymidine. So what is the structure of thymidine? Here, this is the pyrimidine. Pyrimidine, that is the purine. So when we draw, here I draw, it is a single cycle base structure. First we draw the base structure of this is a pyrimidine, hexagonal structure. This side, because it is thymidine, adenosine and amino group is there, so thymidine means double bond O. And we know also here double bond O, this is the nitrogen, first nitrogen, carbon and carbon. 1, 2, 3, 4, this is also carbon. 1, 2, 3 completed, this nitrogen has one H. Now this is also 1, 2, 3 is completed, carbon, 1, 2, 3, 4 is completed, nitrogen, 1, 2, we need one H is here, right? Carbon, we know 1, 2, 3, H, 1, 2, 3, here CH3 is there. Because it is thymine, we can see here, when we consider this one. So 1, 2, 3, 4, 5, 6. Because thymine is also called as 5-methyl. This is methyl group, right? Uracil. Thymidine, right? Okay, this is the nitrogen base. When it binds with pentose sugar, now it becomes nucleoside. Okay, look here how it binds. These are pentose sugar. First carbon, second, third, fourth, and last carbon is outside the same structure which is here, right? This one, H, OH, H, OH. This is the first carbon. Beside draw here OH and H. This first carbon, it has OH group, and this OH group binds with H group, and one H2O water molecule is released, same as to here, right? And this bond, because it is made between first nitrogen and first carbon, this bond is called N-glycosidic bond. One N-glycosidic bond is formed like to this one, same glycosidic bond is formed, and one H2O molecule is released. Their glycosidic bond forms between first nitrogen and first carbon of pentose sugar. Here, N-glycosidic bond is formed between 9th nitrogen of purine and first carbon of pentose sugar. That is the main difference, right? So here we understand how a nucleoside is formed. Okay, a nucleoside means nitrogen base plus pentose sugar, and in between these two, N-glycosidic bond is formed. One water molecule released and one N-glycosidic bond is formed. Now you can draw other nucleosides. Okay,